Hello and welcome. I'm Robert. I'm a registered dietitian specialising in nutrition for weight loss, cardiovascular disease and diabetes. My videos aim to help people live healthier, happier and more sustainable lives. This week, I wanted to explore our current understanding of the physiological controls of appetite and food intake. As I'm sure you know, any excess calories we consume beyond requirements will be stored as fat. Our brains are the primary controller of the amount of food we eat. However, this is also influenced by our endocrine, gastrointestinal, central nervous, peripheral nervous, and cardiovascular systems. Each of these systems can play a significant part in determining our overall weight. This and my next video will focus on the physiology of obesity and the key peripheral and central signals involved in the regulation of our body weight. Homeostatic regulation of body weight occurs primarily in the hypothalamus. We can think of the hypothalamus as the brain's body weight control centre. It collects and processes information about our current satiety levels and fat stores, whilst also considering more complex factors such as emotional and reward factors. Together, these factors regulate our appetite and balance energy in and output. Within the hypothalamus, the arcuate nucleus is the primary region involved in the homeostatic control of food intake. The blood-brain barrier, a highly selective semi-permeable border, does not extend completely around this region of the brain, which allows peripheral signals secreted by the fat cells and pancreas to gain access to the signalling pathway. The hypothalamus also collects signals from the vagus nerve and sympathetic fibres of the nervous system, which report back from the gastrointestinal system via the brainstem. Neurons that receive information from our sensory organs and transmit this input to the central nervous system are called afferent neurons. On the other hand, neurons that carry signals from the central nervous system back to the rest of the body are called efferent neurons. The hypothalamus and brainstem receive afferent signals reporting the current state of energy balance within our internal and external environments. These regions of the brain are then able to process this information to send efferent signals that will promote food-seeking behaviours and also control how our body utilises the food we have consumed. Overall, energy balance will be regulated through short and long-term processes. In today's video, we will look at the two primary hormones that regulate long-term energy balance. These are leptin and insulin. Before we look at these hormones, I have a quick favour to ask. If you found this or other videos I've made helpful, can I ask that you take a moment to support my channel by clicking the like and subscribe buttons. Doing this helps the channel grow, which means I can spend more time making content for you to enjoy. Leptin is a hormone secreted from fat cells that helps to regulate body weight. The name leptin is derived from the Greek word leptos, meaning thin. Discovered in 1994, it helped cement the concept that fat tissue is not an inert store of energy, but a metabolically dynamic organ that can influence the functions of the body. Leptin is able to cross the blood-brain barrier via saturatable transporter mechanisms. If leptin levels are sufficient, it will instruct the arcuate nucleus to reduce food intake. This area of the brain will also increase our energy expenditure by increasing the expression of appetite-suppressing neuropeptides, which promote weight loss and may also stimulate the breakdown of fat and decreasing the expression of appetite-stimulating neuropeptides. Neuropeptides are chemical messengers made up of small chains of amino acids that are synthesized and released by neurons. Although very rare, congenital leptin deficiency can result in severe obesity from a very young age. This is a form of monogenic obesity. I explored this and other genetic causes of obesity in my video, Are My Genes Making Me Fat?, which I'll link below. In humans, leptin deficiency causes a significant increase in appetite, which will result in rapid weight gain. In people living with the condition, leptin treatment effectively reduces appetite and food intake to normal levels, resulting in weight loss. In people with a functional leptin system, circulating levels of leptin are closely correlated with the amount of fat stores within the body. As very low levels of leptin will result in an increase in appetite and food-seeking behaviour, we might expect very high levels to have the opposite effect. Interestingly, we do not see this effect. The failure of high levels of leptin to prevent weight gain can be seen in most people living with obesity, who have both high levels of plasma leptin and strong hunger signals. The development of leptin resistance, which results in reduced leptin transport across the blood-brain barrier, and impaired leptin signalling in the hypothalamus have been hypothesized as potential causes of this observation. As a result, researchers have concluded that leptin primarily acts as part of a negative feedback loop, meaning that when levels drop, we experience a strong desire to find food. But when levels are above a certain threshold, we are not disinhibited from eating any further by having more. This was observed when scientists tried administering exogenous leptin to individuals in the hope of developing it as a weight loss medication. Unfortunately, in those with a functional leptin system, there was no further reduction in appetite. Ultimately, it appears that the leptin system has developed to protect against starvation, a far more likely occurrence during our evolution, than protect against obesity in today's rather unique environment of plenty. Insulin was discovered by Sir Frederick G. Banting, Charles H. Best and J.J.R. MacLeod at the University of Toronto in 1921, and it was subsequently purified by James B. Collip. Many of you will likely be familiar with insulin's main function, which is to regulate the amount of glucose within our blood. However, insulin is also important in the long-term stability of body weight and fat mass. It is produced by the beta cells of the pancreas. Interestingly, insulin has a very short half-life of just 4 to 6 minutes, as it is very quickly removed by the liver and the kidneys. Our brains are able to determine metabolic activity by sensing the minute-by-minute changes in insulin concentrations within our blood. Insulin rises in the blood following a meal in a concentration directly proportional to total body fat and reaches the arcuate nucleus via blood-brain barrier through a saturable transporter mechanism. In rodent studies, we have found that insulin administered directly into the brains causes a dose-dependent reduction in food intake. On the other hand, reducing the amount of insulin in the brain by using a brain-specific insulin receptor knockout results in a significantly increased appetite and weight gain. This research highlights insulin's important contribution to energy homeostasis. In next week's video, we'll continue this topic by exploring the range of short-term satiety signals that regulate our food intake. Anyway, that's it from me. I hope you found this video helpful or informative, and if you did, why not give it a thumbs up and consider subscribing. And before I go, this is your weekly reminder to never stop learning, because life never stops teaching. See you in the next one. Cheerio.